Hello everyone. In this class, we'll try to learn about cell membrane. The membrane that surrounds the cell is a remarkable structure. It is made up of lipids and proteins and is a semi-permeable, allows some substances to pass through it and excluding others. However, its permeability can also be varied because it contains numerous regulated ion channels and other transport proteins that can change the amount of substances moving across it. It generally referred to as a plasma membrane. The nucleus and the other organelles in the cell are bound by similar membranous structures. Although the chemical structures of membranes and their properties vary considerably among one location to another, they have certain common features. They are generally about 7.5 nanometers, that is 75 angstrom in unit. The major lipids are phospholipids such as phosphatidylcholine, phosphatidylserine, phosphatidylethylamine. The shape of the phospholipid molecules reflects its solubility properties. The head end of the molecule contains a phosphate portion and is relatively soluble in water, that is, it is polar and hydrophilic. And the tail ends are relatively insoluble, they are nonpolar and hydrophobic. The possession of both hydrophilic and hydrophobic properties make the lipid an amphipathic molecule. In the membrane, the hydrophilic ends of the molecules are exposed to the aqueous environment that bathes in the exterior of the cells and the aqueous cytoplasm. The hydrophobic ends meet in the water poor interior of the membrane. In prokaryotes, like in bacteria in which there is no nucleus, the membranes are relatively simple. But in eukaryotes, that is the cell which are containing nuclei, the cell membranes contain various sphingolipids, sphingomyelin, and cholesterol in addition to the phospholipids and phosphatidylcholine. Many different proteins are embedded in the membrane. The membrane proteins are classified into two categories, that is integral proteins or intrinsic membrane proteins and peripheral proteins or extrinsic membrane proteins. They exist as a separate globular units and many pass through or are embedded in one leaflet or membrane, that is, for example, integral proteins, whereas peripheral proteins are associated with the inside or outside the cell membrane. The amount of protein varies significantly with the function of the cell but makes up on average 50% of the mass of the membrane. That is, there is about one protein molecule per 50 of much smaller phospholipid molecules. The proteins in the membrane carry out many functions. Some are cell adhesion molecules that anchor cells to their neighbors or to basal laminas. Some proteins functions as pumps relatively or actively transporting ions across the membrane. The proteins functions as carriers, transporting substances down the electrochemical gradients by facilitated diffusion. Still others are ion channels which, when activated, permit the passage of ions into or out of the cell. Proteins in another group function as receptors that bind to ligands or messenger molecules, initiating a physiological change that occurs inside the cell. Proteins also function as enzymes, catalyzing reactions at the surfaces of the membrane. The hydrophobic portion of the proteins are usually located in the interior of the cell membrane, whereas the charged hydrophilic portions are located on the surfaces. Peripheral proteins are attached to the surfaces of the membrane in various ways. One common way of attachment to glycosylated forms of phosphatidylinositol. The proteins held by these glycophosphatidylinositol anchors includes enzymes such as alkaline phosphatase, various antigens, a number of cell adhesion molecules, and proteins that combat cell lysis by complement. Over 250 GPI linked cell surface proteins have been described in humans. Other proteins are lipidated, that is, they have a specific lipids attached to them. Proteins may be myriosylated, palmylated, or phenylated, that is, attached to the guaranal or farnesyl groups. So this is showing a structure of the cell membranes where it is showing the bilipid layer, and outer being the hydrophilic and inside being the hydrophobic. And it is also showing different parts of the cell membrane, that is integral proteins, channel proteins, peripheral proteins inside the cholesterol, and also shows some of the attachments like glycolipids, glycoproteins, globular proteins, and carbohydrates. This is another example of cell membrane where it is showing different types of proteins, so which includes integral proteins and peripheral proteins and some attachments. The protein structure and particularly the enzyme content of biologic membranes varies not only from cell to cell but also within the same cell. For example, some of the enzymes embedded in the cell membranes are different from those mitochondrial membranes. In epithelial cells, the enzymes in the cell membrane on the mucosal surface differ from those in the cell membrane on basal and lateral margins of the cell. That is, the cells are polarized. Such polarization makes a directional transport across epithelia possible. The membranes are dynamic structures and their constituents are being constantly renewed at different rates. Some proteins are anchored to the cytoskeleton, but others move laterally in the membrane. Underlying most cells, a thin fuzzy layer plus some fibrillus that collectively makes up basement membrane, or more properly basal lamina. The basal lamina and more generally the extracellular matrix are made up of many proteins that hold cells together, regulate their development, and determine their growth. These include collagens, laminins, fibronectins, tenasins, and various proteoglycans. These are some of the functions of cell membrane because of its structure. They are protective in function, they have very selective permeability, they have absorptive function, they have excretory function, they are important for exchange of gases. They also help in the maintenance of shape and size of the cell. Thank you.